All right. Hi. Thank you all for being here. I'd like to talk to you today about fine roots resource strategies along an environmental gradient in Western Canada. And I've been working on fine root traits and long-term changes in climate. First, because fine root traits drive ecosystem processes such as carbon and nutrient cycling. So knowing how plants adjust their below-ground traits to long-term changes in climate, it's important because it may have significant outcome for tree persistence and resilience in our changing climate. So let's start all on the same page. Brief description of fine roots, and especially tree fine roots. They are the most terminal roots within the root system hierarchy. And they are mainly responsible for soil resource uptake. They are the most dynamic and physiologically active part of the plant below ground, and their production and turnover represent about 22% of the terrestrial net primary production globally. So depending on their location within the root system and depending on the degree of their secondary growth, fine roots may have different function and characteristic. And for this project, I have been focusing on the absorptive pool of fine roots, which are the fine roots that are mostly responsible for soil resource uptake. And they are also classified as usually the first three root orders. So one approach to determine fine roots resource strategies is to look at their functional traits. So fine roots functional traits are basically plant traits. So they are a measurable aspect of a plant, they characterize the species of interest, and they impact plant growth, reproduction, or survival. So I have been focusing on three main fine roots functional trait groups: morphological, chemical, and architectural. And for morphological and chemical traits, I have been measuring traits that relate to trade-off between root constriction cost and resource acquisition. So for example, root diameter, root tissue density, specific root area and specific root length, root carbon to nitrogen ratio, and nitrogen content. And for architectural traits, I've been mainly focusing on branching intensity and branching ratio, which usually relate to the way the roots intercept and explore the soil, intercept the resources from the soil and explore the soil. All right, so I've been focusing on those traits, and my first objective was to quantify root traits dimensionality, which was one of the research frontier highlighted in 2016. And the first hypothesis I had was that each of the root traits group I'm looking at will respond independently and differently to long-term changes in climate. So to test this hypothesis, I've been working on the widely distributed Douglas fir tree species. And this Douglas fir tree species is a very good model species because it has a wide geographical range. Here I'm just showing you the range in southern British Columbia, but it also goes down to New Mexico, or no, actual Mexico. So wide geographical range. It is known to be locally adapted to climate for above-ground traits, such as growth and bud phenology. So here you have the mapped genetic variation in growth potential of Douglas fir. So each of the different colors represent different growth potential, so potentially different tree population. But what we don't know is this variability that we observe for above-ground traits would be mirrored for below-ground traits. And so to tackle this question, I've selected a strong climate gradient within this constrained geographical area. So this is southern British Columbia. And the gradient was from like about 49 degree north to 52 degree north. And as you go from the south to the north of the gradient, the climate gets drier, colder, the soil gets poorer in phosphorus, but richer in nitrogen, and the soil is characterized by higher cation exchange capacity. So the gradient is pretty short compared to what we can see in other papers, but it also allowed me to minimize the differences in day length among study location. So for each study location that you see here, I had 3 independent study sites. I targeted 5 Douglas fir tree per site, and I extracted 1 soil blocks per tree, like it was 20 centimeter by 20 centimeter by 20 centimeter. And so in total, I extracted 365 fine root branches from 75 soil blocks. All right, so what about fine roots dimensionality? Here, I'm showing you the results from the principal component analysis. And the 2 principal components represented about 50% of the data variation. And the study location are color-coded. So the first thing to note from this graph is the first axis of variation, mainly represented by root diameter, specific root area, and specific root length. Let's see if I can see. Okay, so root diameter, specific root area, specific root length. Root carbon, some people would argue that's not really a root trait. All right, and this axis of variation was quite independent from the other axis of variation. Oh, what am I doing? Sorry. Okay. Okay, so it was also independent from the second axis of variation, which was related to root architectural traits, so branching ratio and branching intensity. And those 2 axes were also quite independent from root tissue density and root nitrogen content, which represented like kind of a third axis of variation. So with these results, we show clear multidimensionality across within species for fine root traits, and explicitly accounting for this functional dimension should aid in developing a more realistic fine root traits framework. So the fact that root traits are dimensional was also demonstrated across species, and I'm showing you the results from the study of Ma et al. They also show that across 7 biomes, specific root length, root tissue density, and root nitrogen were quite independent. So that means that among and within species, multiple trade-offs among root traits may be effective in enabling plants to cope with diverse environmental limitations. All right, so the second objective of this project was to determine trait-environment linkages. And for that, the hypothesis I had was that fine root traits in harsher environments, so colder, drier, or with poorer soil, would favor greater acquisitive capacity. So that means that the fine root traits would represent an acquisitive strategy for resources. And that should translate in having thinner fine roots with higher specific root length or root area, lower tissue density, lower carbon to nitrogen ratio, so higher nitrogen content, and higher branching intensity or branching ratio. Right, so first results here, I'm showing you a graph for root tissue density and mean annual temperature, and the first 3 root order are color-coded, and those lines were fitted with linear mixed effect model with loctane transform data. So what we observed first with root tissue density is that it increased with temperature by about 4% per degree for all the root order considered. Same thing for root carbon to nitrogen ratio. It also increased with temperature by about 1% per degree, but only so that was stronger only for the second and third order roots. Alternatively, specific root area and root diameter decrease with temperature. So here, this is the results for root diameter. It decreased by 2% per degree, and that was especially significant for the third order roots. And specific root area also decreased with temperature by about 3% per degree, especially for the first and second order roots. So because we see clear relationship between climate and in this case, mean annual temperature and fine root traits, it may, within species, it may impact below ground processes with important outcomes for tree persistence and resilience. So you might wonder about root architecture. Well, it was weakly associated with soil fertility. So here I'm just showing you root branching intensity and soil available phosphorus. And we just noticed a slight increase in branching intensity with soil available phosphorus, but that was barely significant. And so, all right, so the initial hypothesis was that fine root traits in harsher environment would favor greater acquisitive capacity. So to address this hypothesis, I'd like to use the root resource strategic framework adapted from Wemstra. So all of the root traits that fall into this part of the diagram represent an acquisitive strategy for resources. And if they fall into this part of the diagram, they would represent a conservative strategy for resources. And as you can see, there are physiological root traits that I haven't measured, and they are in gray. All right, so in harsher environment, what I observed is that the specific root area was higher. I didn't see any relationship between specific root length and environmental variables. Higher nitrogen content, higher root diameter, lower root tissue density, and lower carbon to nitrogen ratio. So from this diagram, I could conclude that the hypothesis is partially validated. But what we also would need to know is how, because these changes alter resource allocation and nutrient cycling throughout the ecosystem. And I'd like to go back to the root diameter as being a conservative trait. And one could argue that higher root diameter could be an acquisitive trait if it's related to higher mycorrhizal fungal colonization, because it would increase the resource acquisition potential. So interestingly, I didn't find any differences in fungal colonization across the gradient. So the colonization rate was about 95% for all the fine roots. But I would suspect that root diameter might be or might relate to other fungal traits, such as the exploration type. So here I'm showing you two different type of exploration type, so medium distance with the fungal species Piloderma, and short distance with Lactarius. So this is what I'm working on right now, trying to see if there is any relationship between fine root traits and these mycorrhizal traits, so stay tuned. All right, so the take-home, our studies show multiple fine root traits, or we provide evidence for multiple fine root traits dimension. We show that intraspecific fine root traits variability relate to climate, and we also provide some evidence that fine roots increase their potential to acquire resources to adjust to harsher environment. So I'd like to thank all of the great people that have been working with me in the field and in the lab, and thank you all for your attention.